Um, it's so good to be back. I have a soft corner for RSG, and I'm really happy that we're back and meeting for many of us in person. So I'm excited to present today some interesting ongoing work being led by my PhD student, Payam Divania, who couldn't present it himself because of some unavoidable conflicts. So as you all know, machine learning models are making great strides in regulatory genomics, and feature importance scores have played a very useful role for interpretability of these models. For instance, leading methods for gene regulatory network reconstruction, or GRN reconstruction, use feature importance of ML models such as random forests to select transcription factors regulating a gene. In this project, Payam decided to look deeper into whether there's a causal or mechanistic interpretation of such feature importance scores. And additionally, he wanted to test if change in feature importance between two data sets or two conditions or populations can reveal differences in the underlying GRNs. So let me elaborate on this last point about differences in GRNs. Let's say we have expression data from two conditions or populations with corresponding GRNs being shown on the right. And the goal is to find cases like these where the GRNs differ. In other words, given expression data from two conditions, say case and control, we'd like a method to produce the differential edges, which I'll call the DGRN. A whole family of approaches to this problem, in fact, the more successful ones, infer the strength of each regulatory association based on co-expression, and then devise tests for significant differences thereof between the conditions. There are also methods that adopt joint modeling of the two data sets in order to detect DGRN edges. An alternative approach that is surprisingly less explored is the following. We know that widely popular GRN reconstruction methods such as Gini3 and NNet model target genes expression as a function of all TFs and use feature importance to score TF gene relationships. So it would seem natural to ask if we can use the difference in these feature importance scores to score or rank DGRN edges. Such an idea has been explored to some extent in the context of linear models where the coefficients provide the feature importances. In this talk, I will present Payam's method called SIMLA, motivated by these ideas. So SIMLA models the target gene in terms of TFs using machine learning models such as random forest or neural network, and then uses the SHAP score of Lundberg and Lee for feature importance and directly compares these SHAP scores between the two data sets or models. Along the way, I'll try to present the SHAP score as a causal or mechanistic measure of TF influence on a gene. I will show some performance evaluations and a case study on a real data set. So let's talk about that causal interpretation. Say we have candidate transcription factors 1 through M, and XT represents the expression of TFT, while Y is the target gene expression. The causal definition of the TF's influence is the average treatment effect, or ATE, defined using Pearl's do operator as the expected gene expression if we set the TF level to on minus the expected gene expression if we set the TF to off. This is loosely speaking how a biologist might think of a TF-driven regulation. One problem here is that setting a TF to on or off may lead to other TFs changing as well, and an indirect effect may be registered as a causal influence. Partly motivated by this, we next define a local version of the ATE called the LTE, quantifying the effect of XT, the transcription factor T, on the target gene Y for a particular sample X. This looks a bit like the ATE, but note that we now force the other TFs to stay where they are in the sample X and compare the effect of setting XT to on versus off. I admit this gets into the realm of fantasy so far as experimental interventions go, but please indulge me in this thought experiment. Another problem still lurking here is the binary treatment variable to represent the regulatory action of a TF whose effects are concentration dependent and often nonlinear. So the next modification is to parameterize the local treatment effect, or LTE,
with a constant xt hat that represents a suitable baseline level of the TF, and comparing the effect of setting xt to what is seen in the sample X to the effect of setting xt to the assumed baseline level xt hat. The LTE parameterized by this baseline level can then be averaged over some distribution of that baseline level to obtain the ultimate LTE. All this has been just playing with definitions, using causal language to help think mechanistically about the effect of a TF on the target gene in a specific sample X. Now we move over to observational data with the goal of estimating this local treatment effect, or LTE. First, as you might expect, we train an ML model to predict the target gene's expression using TF expression. Then we use the local SHAP score to quantify the contribution of the XT, each transcription factor T, to the model's output for the sample X. And that's it, because we proved that this local SHAP score is an estimate of the LTE we defined in the previous slides. The proof works under the assumption of a causal diagram shown here, where the TF of interest, XT, has a causal association with Y, and we seek to quantify this association. The other TFs, XIs, may or may not be associated with Y, and in this sense, the causal diagram is not fully resolved. Our solution to this incomplete causal diagram is to average the LTE over all possible causal structures consistent with this diagram. And this average is what we find to be equal to the SHAP score, the local SHAP score, under certain assumptions. So now, having gained a mechanistic or causal understanding of the SHAP feature importance, we are ready to go back to the DGRN problem with this simple algorithm. Train a machine learning model FC on the target gene for each of the two conditions or populations denoted by C. For each sample, calculate the LTE of transcription factor T in each model using the SHAP score. Take the difference of LTE values on the same sample under the two models. Calculate the root mean squared of the LTE values over all the samples in the treatment group. And that's the similar score of differential importance of the transcription factor T for the target gene. So I'll quickly describe some of the evaluations of the method on synthetic data. So we used our GRN-driven single-cell expression simulator Sergio to generate 15 data sets, each comprising a pair of GRNs and their corresponding expression matrices. Sergio samples cells from a dynamical process describing gene expression with biological noise. And the pair of GRNs in each data set share between 45 to 95% of their edges. The expression data comprises 3,000 cells over 10 different cell types in each condition. We evaluated SIMLA as well as several differential co-expression methods selected as being top performers in a benchmarking study from 2019. We also evaluated Gini3-based DGRN prediction, since Gini3 is one of the most popular GRN reconstruction methods, as well as a differential regression method called BOOSTIF, that's on bioRxiv. In the first simplest evaluations, we found the AUROCs and the normalized AUPRCs of the different methods to be comparable. But then we moved on to the more challenging scenario where master regulators of the GRN can themselves be differentially expressed between the case and control conditions. That is, there is a strong confounding effect from the biological condition. Now the performance of the differential co-expression methods dropped dramatically, while similar performance remained largely unchanged. We did the same comparison to an approach based on Gini3 feature importance scores being compared between the two data sets, and the result was significantly worse. However, when we redefined the differential importance based on ranks of Gini3 scores within each condition, the Gini3 based approach was similar to, although somewhat worse than SIMLA. Note that this alternative method, also based on comparison of feature importance scores, does not have the nice mechanistic interpretation that we found for the SHAP scores. We repeated the evaluations with varying levels of dropout added to the data and found that SIMLA, now tested with two settings, one with random forest and the other with an MLP as the underlying ML model, to be clearly better than the compared methods for all levels of technical noise. And assured by these extensive evaluations on synthetic data, we finally used SIMLA to characterize the differential regulation underlying Alzheimer's disease, analyzing single-cell RNA-seq data from 22 individuals in each of two groups based on diagnosis at the time of death. we combined similar derived DGRN information with a cis-regulatory GRN for multiple psychiatric disorders as derived by the Psych and Code Consortium. And this figure summarizes the transcription factors identified by this integrative analysis, with the size of the circles representing the Regulon size and the two axes representing the strength of evidence from SIMLA and the Psych and Code. We are currently examining this network for new biological insights. And that's all I have to say. I would like to thank my funding agency, and thanks very much for listening. Happy to take questions. It was red then.
Okay, hi. I'm just wondering about how your approach might handle redundancy in the regulatory network, like how the importance difference would be there or not if the underlying machine learning model has learned one edge in one condition and another in the other condition.
That's right. So in the theoretical treatment, the machine learning model is assumed to be accurate, and we don't have. That's one thing. It doesn't work if the machine learning models are not accurate. I think what you're talking about is like multicollinearity or redundancy of features.
Redundancy of the factors, yeah.
We don't have a great way to address that except to empirically examine the results. So no good answer to the redundancy of features.
Okay, thanks.
Thanks for a great talk. A question I had is, you're using transcription factor expression to predict gene expression. Oftentimes you see a low expression of a transcription factor and it still can enable high expression of its target gene. And there's a lot of new data that has paired RNA-seq and ATAC-seq or some other assay for enhanced activity. Have you thought about incorporating, for example, transcription factor motifs in nearby open chromatin regions into your approach?
Absolutely. So fortunately, there are many people working on exactly that, and we have also tried to work on that problem. This is examining just one aspect of the analysis where we're trying to relate things based on expression only. It can be useful when someone does not have the cis-regulatory information, but naturally, future work should try to integrate cis-regulatory information for more mechanistic constraints.
Thanks.
Hi, fantastic talk. My question is like, you presented a beautiful work on the case control comparison scenario. I was wondering if your method is generalizable for like a continuous label, like for example, the disease severity types of label, and try to infer the regulatory network underlying those continuous labels of the samples.
So the outcome variable Y is continuous. And in fact, one of the challenges we faced was that the feature itself, the treatment variable, which is typically binary in causal inference settings, had to be continuous. And then it went into the issue. So it is continuous already. I don't understand.
I was trying to ask like whether case control, so the treatment variable, maybe I.
Oh, okay. So the case control is currently treated. It's not treated as a variable in the causal diagram. We'd like to, and I did not present that yet, but that is not the treatment variable. The treatment variable here is the transcription factor's presence.
Sorry, I think.
Yeah. No, I know it's an unusual setting.
Thank you.
I just want to ask one quick question. How would it work across multiple conditions? So if you have like
I haven't thought of it. Great question. Yeah.